So we have question number 1 here. We have an investigator who is studying bacterial adaptations to the environment in an infectious isolate and a non-infectious isolate of Haemophilus influenza type B. Now, animals are exposed to the non-infectious isolate do not develop symptoms for the infection. Now, the investigator cultivates non-infectious isolate in a culture with lysed bacteria from the infectious isolate. Some of the animals exposed to these bacteria develop symptoms of infection. This is a basic microbiology questions. It's your first chapter in a microbiology textbook is this. So is it transposition or transduction, or is it a conjugation or a reassortment, transformation, or generalized transduction? Okay, now what is transformation? It's a process in which transforming the bacteria transforms, as in what? It takes up your naked DNA from lysed bacteria or the surroundings through what? The cell membrane, and that will incorporate the DNA into their genome. And once you have new DNA, you will express new genes. Several species can do this process, like pneumonia, Neisseria, Hib, and all of that. That's called transformation. So just try to remember, it's a new word and you can't remember, you got confused and you marked something else. Transformation is the bacteria becomes a new one, it transforms. Okay, transposition is in which DNA from one location in the genome is excised and then reintegrated into a different location. It's like we're doing graft in plastic surgery. That this process can transfer genes between plasmids and chromosomes. It does not describe the transfer of genetic material from one bacterial to the next in the serial experiment. Like, it's just in that particular genome there's a difference. It is not going to be picked up by the other empty guy. Okay, that's what they're telling. Specialized transduction is a process in which bacterial DNA is transferred from one bacterium to the another via something. It's a bacteriophage. It's not like something is in the environment and I directly pick it. That is transformation. Transduction is when they pick up through bacteriophage. Now, a lysogenic bacteriophage infects a bacterium and inserts viral DNA into the bacterial chromosome. Now, when this DNA is excised to make new viral particles nearby the bacterial genes are incorporated into the bacteriophage cascade. This bacteriophage then lyses the infected bacterium and can transfer fragments of this genetic material when it infects a new bacterium. Bacteriophages are not used in the experiment. Remember this. Now, what is conjugation? It's in which we know plasma F, a plasmid from fertile to non-fertile through sex pillars, and they transfer genetic material. That's it. That's all you need to know here. And option D is genetic reassortment occurs with viruses, not bacteria. Viral reassortment can occur when two or more distinct viral strains with segmented genomes co-infect one cell. New viral particles are assembled with genetic segments that originated from different viral strains. Thus, they contain significantly different combinations of genes from the original viral strains. What is option F? It was generalized transduction, in which a bacterial DNA is transferred from again one to the other to your phage. A lytic can infects a bacterium and cleaves bacterial DNA. It becomes packaged in the phage cascade. Now, when the phage then infects another, this can transfer the DNA, and you cannot use bacteriophage in the experiment. Okay.
Thanks for watching. Please give a thumbs up and subscribe for more.